Hello everyone and welcome to Abhith Siksha. This is class 9 science and we are doing the chapter the fundamental unit of life which falls under biology. So this is part 5. So do you remember what we did last class? Let us do a quick summary before starting our today's class. Last class we studied about cytoplasm and the cell organelles. So what is cytoplasm? If I draw a general representation of a cell, okay, if I draw a general cell, then the outer layer, the covering of the cell is our cell membrane or the plasma membrane. Yes, so this is our plasma membrane. In the center of the cell we will find the nucleus. So let me write N over here, and the space between the plasma membrane and the nucleus, all this space, what is this space filled up with? This space is filled up with cytoplasm. Yes, the fluid content inside the plasma membrane is called cytoplasm, and in the cytoplasm we have a lot of cell organelles floating in it. So what are cell organelles? Cell organelles are membrane bound little organs of the cell. Under cell organelles, I told you that we need to study 6 cell organelles for our class 9. And under that, our first cell organelle was the endoplasmic reticulum, in short ER. So what is ER? ER are large network of membrane bound tubes and sheets. And then we studied that there are 2 types of ER, that is RER and SER. R for rough, S for smooth. Why RER is rough? Because of the presence of ribosomes. What do ribosomes make? They make proteins. What do SER makes? SER makes lipids or fat molecules. Then we studied that they serve as a transport channel, and they function as the cytoplasmic framework. So this was everything that we studied last class. In our today's class, we will be studying these. In our today's class, we will be studying Golgi Apparatus. Then under that, we need to study about Golgi, structure and location, meaning where the Golgi apparatus is located inside the cell, the functions. Then number 2, we have lysosomes, the structure and location of lysosome, and then lastly we need to study the functions of lysosomes. So let us start with Golgi apparatus. Golgi apparatus. So Golgi apparatus is a cell organelle, meaning it is a little organ of the cell floating in the cytoplasm, and Golgi apparatus was first described by Golgi. This person over here, he described Golgi apparatus for the first time. Now, who is Golgi? There is a little paragraph on your book. Since it is there, we need to study that. Okay, questions may come from that section, may not come from that section, but since that part is there, let us study a little bit about Golgi. So, about Golgi. He is the first person who described Golgi apparatus. He was born in July 1843 in Cortino, and Cortino is a small village in the northern Italy. Okay, he was born in 1843, Cortino, and Cortino is a small village in northern Italy. Golgi was the chief medical officer at the hospital of chronically ill. Okay, and then in that particular hospital, which hospital? Hospital of chronically ill. In that particular hospital, he transformed a little kitchen. He selected a little kitchen of that hospital and he transformed that little kitchen into a rudimentary laboratory, into his own personal rudimentary laboratory. Rudimentary meaning basic. Rudimentary, in short, we can say a basic laboratory which does not have much facilities. It has only the basic facilities. So he converted a small kitchen of this hospital into his small personal rudimentary laboratory, and in this laboratory he performed various experiments, he performed various studies, and he devoted his life towards science in this little kitchen laboratory of his own. Okay, now let us learn more about him. One of his revolutionary works, he did many works, okay, he have given a lot in the field of science, but one of his revolutionary work is a staining method known as black reaction. So there is a reaction, black reaction. This black reaction is a staining method. See, it is a method of staining with a weak solution of silver nitrate. the solution that is used, the staining solution that is used, it is a weak solution of silver nitrate, and the entire staining method is known as black reaction. Okay, here I even added a picture. See, black reaction. This is how after the staining the specimen looks like. This is after staining. Okay, let us look at the next point. Okay, at this point let me add that Mr. Golgi, he was very much interested in the nervous system. Okay, nervous system of animals, nervous system of humans, as in all, he was very interested in the nervous system. So major part of his studies, major part of his experiment, major part of black reaction he conducted on nervous system. And it was in the nervous system, it was in the nerve cells that he first noticed Golgi apparatus or Golgi bodies. Okay. He also shared the 1906 Nobel Prize with Santiago Ramon y Cajal for their work on the structure of the nervous system. See, I told you, right, that he was very much interested in the nervous system. So, because of his study on the structure of the nervous system, he even received the Nobel Prize in the year 1906 with another scientist whose name is Santiago Ramon y Cajal. Okay, so this is almost everything about Camillo Golgi. But since our main topic is not Camillo Golgi, our main topic of discussion is Golgi apparatus. So now let us start about the Golgi apparatus or Golgi body. Whichever you prefer, you can say that. So Golgi apparatus, first let us study about the structure and location. How does Golgi apparatus looks like? How does Golgi body looks like? And where is it located inside the cell? So Golgi apparatus, it meaning Golgi apparatus. Golgi apparatus consists of a system of membranes. It is a system of membranes. We have a lot of membranes, and these membranes, they are stacked up. Okay, let's read. It consists of a system of membranes arranged approximately parallel to each other in stacks. It is, we have membranes. Okay, the membranes are stacked up, they are piled up. Okay, how are they piled up? They are piled up parallel to one another. System of membranes approximately parallel to each other, and they are piled up, they are stacked up, and these stacks are known as cisterns. They are piled up or they are stacked up. These stacks are known as cisterns, along with some membrane bound vesicles. We will find this stack of membrane. Along this stack of membrane, we will find some membrane bound vesicles. What do we mean by vesicles? Vesicles, in short, I can say that vesicles are sac-like structures, mostly filled up with fluid or liquid. Okay, so along with membrane bound vesicles. They are mostly found near the nucleus, and the membranes often have connections with the membranes of ear. So where are they found? They are found mostly near the nucleus. Okay, mostly they are found near the nucleus, and the membranes often have connections with the membranes of ear. Okay, why do they have connections with the membrane of ear? Okay, let me first ask you, what does ER manufactures or what does ER synthesizes? Last class we discussed about ER, that is endoplasmic reticulum. What do ER makes? ER makes proteins and lipids. ER makes proteins and lipids. One is made by RER, the other is made by SER. Let us not be that specific. As in all, we can say that ER makes proteins and lipids. Okay, these proteins and lipids, they are synthesized in the ER. ER only makes them. Okay, these proteins and lipids, what happens is that they are packed up. Okay, they are packed up, and this packed up lipids and proteins, these packed up little balls are taken by the vesicles to the Golgi apparatus. Vesicles take up this packed up proteins and lipids to the Golgi apparatus. Inside the Golgi apparatus, what happens? Transformation takes place, modification takes place. After modification, the vesicles will again take up the modified substance. it will again take up the modified complex, the modified substance, and the vesicles, what they will do, they will either take this modified substance to the plasma membrane or the cell membrane. It will either take the substance there, or it will take the substance to some other cell organelles. Okay, so this is about the structure and location. Let us learn about its function. Functions. The major function of Golgi body is to package. See, I told you that all the protein and lipids, they are synthesized or they are manufactured in the ER. The protein and lipid, what will happen? It will get packed up. This packed up little balls will be taken by vesicles to the Golgi body. So, major function is package, modify. Where do the vesicles take this packed up balls of proteins and lipids? To the Golgi apparatus. There modification takes place. Modifications like how? Modification, for example, simple sugar will make up complex sugars. Okay, such kind of modifications will take place. And then after modification, the vesicle will again take this modified substance either to the plasma membrane or the cell membrane or to some other cell organelle. So, and transport. Various materials synthesized near the ER. So, all these statements, they are aligning, they are making sense now. Okay. So, finally we can say that the major function is what? Packaging, modification and transport. Okay. Then it also involves in the formation of lysosomes. Golgi apparatus, they are involved in the formation of lysosomes. Now this is a new word. What is lysosomes? What is this lysosomes? Lysosome is a cell organelle. Okay, it is a cell organelle, and we will discuss about lysosome just after Golgi apparatus. Okay, once we finish Golgi apparatus, we will start our lysosome. Okay, so it is also involved in the formation of another cell organelle whose name is lysosomes. So the number of Golgi apparatus may differ from cell to cell, but in every cell where Golgi apparatus is present, it performs a very important function. Which functions? These three functions and the formation of lysosomes. Okay. Now let us move on to lysosomes. What is lysosome? It is a cell organelle. Where is lysosome made? It is made in the Golgi apparatus. How do we know that? Just in the previous slide we were discussing that. So we know that Golgi apparatus is used or it is involved in the formation of lysosomes. Now, the structure and location of lysosome. How do lysosomes look like? Where is it located inside the cell? Lysosomes are the waste disposal system of the cell. Lysosomes are the waste disposal system. In a cell, many kind of reactions, many types of processes takes place. Okay, there is a lot of work going on inside a cell. So inside the cell, a lot of material is broken down into simple products. A lot of simple products are clubbed up together and a complex substance is made. So a lot of processes are going on. All the things that are made inside the cell, among all of that, some are very important that the cell needs, some are very useless, or we can say that they are waste matter. The cell does not need any waste matter. Okay, so if there is waste inside the cell, will the cell cherish that waste product and keep it close to its heart, or will it try to throw it? The cell does not need the waste product. So what the cell will do? The cell will try to throw away the waste product because it's useless. What will the cell do with the waste, right? So it will try to throw away. All this waste disposal process, the cleaning of the cell, this process is done by lysosomes. How do lysosomes do that? Okay, first of all, let us study the structure. Okay, so they are the waste disposal system of the cell. They are membrane bound sacs. Lysosomes, they are membrane bound and they are sacs. Sacs meaning they are like little balls and there is something inside that bag. Okay, they are like little bags or little balls, and inside that ball, inside that bag, it is filled up with something. What is it filled up with? They are membrane bound sacs filled with digestive enzymes. These little bags or this little ball, it is filled up with digestive enzymes, and these digestive enzymes are made up by RER. RER meaning rough endoplasmic reticulum. The digestive enzymes are made up by rough endoplasmic reticulum. These digestive enzymes, these enzymes are very powerful. Okay, they digest meaning they break down substances. What do digestion leads to? Digestion leads to breaking down of complex things. So these digestive enzymes, what they do, they digest substances like any bacteria that the cell does not need, any other thing that the cell does not need, waste for example, or any dead cell organelle inside the cell that the cell does not need. All these things are digested or they are broken up by the lysosome. Okay, now let us move. Okay, before moving on towards the function, this is a cell. So can you spot the lysosome? Lysosomes are mostly spherical in shape, mostly. Okay, they are mostly spherical in shape, they are membrane bound. So can you see this spherical little sacs or bags? So these are lysosomes, and inside the lysosomes we will find enzymes, digestive enzymes made up by ER, endoplasmic reticulum. Which ER? Rough endoplasmic reticulum. Moving on towards its function. The digestive enzymes of the lysosomes. Lysosomes have digestive enzyme. The digestive enzyme of the lysosomes digest foreign materials. They digest foreign materials and worn out cell organelles. I already told you that foreign materials like bacteria or any other thing that the cell does not like or does not want, all those things, and worn out cell organelles. Inside the cell, if there are cell organelles which are dead, useless or worn out, so all those cell organelles also the lysosomes, the digestive enzymes of the lysosome will digest or will break down. They help in keeping the cell clean. They help in keeping the cell clean. Okay, there is nothing to explain here since it is removing all the waste, so it is helping the cell to be clean. During disturbance in the cellular metabolism, lysosomes can also digest their own cell. What happens when a cell gets old or when a cell is unable to perform to its full extent? Okay, when the cell is not doing very well, then there are disturbances in the cellular metabolism. There are disturbances in the cell processes, and when these disturbances take place, what the lysosome does is that the lysosome bursts. Okay, this is our lysosome. Inside the lysosome we have a lot of digestive enzymes, These are the digestive enzymes. When a cell is unable to perform to its full extent, when the cell is old, when the cell is dying, so under that condition, what happens? This lysosome will burst. When this lysosome will burst, all the digestive enzymes that is inside the lysosome, it will spill out. And it will fill up the cytoplasm. So now in the cytoplasm, if this is our cell, so now in the cytoplasm we will find digestive enzymes, and these digestive enzymes are so strong that they have the ability to digest its parent cell as well. So these digestive enzymes, what it will do? It will digest its own cell, its own parent cell. Okay, due to this reason, let me rub this part first of all. Due to this reason, lysosomes are also known as suicide bag of the cell. Why? Because it is like a suicide mission. Lysosomes are capable of killing itself meaning it is capable of killing its parent cell. So that is why it is also known as the suicide bag of the cell. So we can conclude lysosomes to be, they are sac-like structures, they are membrane bound sac-like structures. Inside we have digestive enzymes. These digestive enzymes are so powerful that they can eat up their own parent cell as well, and the main function is it digests foreign materials, help to keep the cell clean. Okay, so this is all about lysosomes, and that was all about Golgi apparatus that we studied. Okay, this is all that we will be studying in our today's class. Before wrapping up our today's class, let us do a quick summary about the things that we learned in this class. So number 1, Golgi apparatus consists of a system of membranes. We already studied that. What is Golgi apparatus? A system of membranes arranged in stacks. What are these stacks called? The stacks are called cisterns. How are they arranged? Parallel to one another. Along with vesicles near the ER. Vesicles meaning little sacs. Sacs which mostly contain fluids. They pack, this is the function, okay, this is the function. They packs, modifies and transports various substances and also make lysosomes. What are lysosomes? Lysosomes are again yet another cell organelle. Lysosomes are waste disposal system of the cell which can digest almost all organic matter. It can digest almost all organic matter including the parent cell, and helps keep the cell clean. They are also known as the suicide bags of the cell. See, suicide bags of the cell. Why suicide bags? Because it's like a suicide mission. If the cell metabolism gets disturbed, the lysosome will burst, the digestive enzyme will fill up the cytoplasm. As a result, this cell, the parent cell, will be eaten up, will be digested. Okay, that is why it is known as the suicide bag of the cell. So this was all for today's class. We learned about Golgi apparatus, we learned about lysosomes. There are more cell organelles to be learned, okay, and all those cell organelles will be continuing in our next class. This is all for today. Thank you for joining and I will see you in the next.